everyone. Good morning or good afternoon, depending on from which location you are joining us today. My name is Helena Kandarova, and I'm the president of STIF and board member of ASCCT. And today it's my big pleasure to be a moderator of this special section or the special webinar that will be on the theme Science for Policy and Policy for Science. And as you know, it will be organized to support the people of Ukraine. From this place, I really would like to thank you for your kind contributions that were collected with the help of ASCCT and that will be sent to the charitable organization People in Need after this webinar. As announced, this webinar will discuss the impact of science on policymakers and provide advice on how to bring science to their attention. The webinar is dedicated especially to the researchers and research organizations that are aiming to achieve policy impact. We have today with us three excellent speakers, Dr. Kristie Sullivan from PCRM, François Busquet from Attendees, and Nataliia Bubalo from National University of Food Technologies, Ukraine. As usual, this webinar will be recorded, and in a few days it will be posted into the archives of our web pages. And if you would like to ask any questions to our speakers, please type it into the Q&A area. There will be about 5-minute Q&A after each of the talk. So I encourage you to type in your questions while they are speaking, and we will try to answer all your questions with the speakers. As you know, we are using closed captioning subtitles. So if you would like to enable it or hide it, you can do it in the bottom of the toolbar where you see the live transcript. And finally, I encourage you to say hello to each other via the chat. Before we start, I just would like to give a quick note about some upcoming events and activities. I will share my screen with you so that you can see what's coming up. So first of the activities that might be of your interest is the upcoming webinar. The registration is open for the next session of the NURAS webinar series. The future is already here, or is it? The meeting is open for the registration and will be streamed on July 8, 2022. I invite you to follow the ASCCT webpage for further details on this. Our colleagues from ASCCT are moving ahead with the planning of the 11th annual meeting that will be held in the Research Triangle Park in October 19 to 21 in New Carolina. Abstract submission is open, and I encourage you to submit your science into this meeting, into the highlighted topics, or in any other relevant topics that might be within the scope of this meeting. From our end, from Europe, I would like to update you about the organization of the 21st International Congress in Barcelona, Fiji, in November 21 to 25. Our submission, abstract submission is cancelled. It's closed, sorry, not cancelled, it's closed already. But if you would like to submit abstracts for the poster presentation, we will again open abstract submission probably in August. And you can have a look into our webpage about the themes and topics that might be relevant for you. We are looking forward to many interesting talks and speakers from the whole world. Last but not least, let me invite you also to our upcoming webinar in July that will be on the guidance and standards for increasing regulatory confidence in vitro and PBK modeling. This webinar will be delivered by Alicia Paini from ESQ Labs and by Amanda Auri from IIBS. Now it's time to proceed to the lectures that are ahead of us. And I would like to invite our first speaker, Dr. François Busquet. He is a French toxicologist with a doctoral degree in life sciences. He obtained his doctoral degree at Technical University Dresden, Germany, and he has master's degree of science in biotechnology obtained in Bordeaux, France. Since 2012, he founded Attendees to advocate 3 Rs to the members of the European Parliament and expanded Attendees activities to educations and trainings, consulting, communication, and dissemination of scientific results to targeted audiences, and in particular policymakers, peers, and general public. François, the floor is yours.
Thank you, Elena. So here I'm sharing my slides and going in presentation mode. I guess you all see it. I guess it's a yes. So I'm gonna share with you today more than almost 10 years of experience working in Brussels with policymakers, mostly through the eyes of CAT Europe, and presented as an honest broker. So briefly, what CAT is doing with the policy program: we are informing the policymakers through evidence by organizing workshops or meetings with different political groups on specific topics regarding triage. We are also a stakeholder platform for the alternative to animal testing, and we have regulatory authorities and NGOs also part of the board. But we also generate independent reproducible research by generating our own data and knowledge, but also participation in EU projects such as Risk Hunter for CAT Europe. So that's in a nutshell how I present the university in Brussels. The general way I interact with policymakers are at the end of the day in three ways. Either you meet them whenever there is a policy agenda that meets the CAT Europe expertise. For example, we are talking now a lot about the CLP, so the classification, labeling and packaging, or the REACH revision. We had in the past also the chemical strategy for sustainability. These are opportunities where you discuss with the policymakers. So you can suggest amendments, so modification of the original text, or you can help them draft written questions directly to the Commission when you are talking about elected policymakers, so politicians. You can also disseminate the information on alternative and research and development when it's not yet the time to discuss a file. That allows you also to have slowly more opportunities to explain omics, grouping, Red Cross, AOPs, so all our jargons that is quite difficult to grasp within a 20 minutes appointments with an MEP or an assistant. So it helps build a relationship and the knowledge around the topic. The other way, or the last way so far I have identified, is to, when you have a blind spot in the policy pipeline, and it is also outside the political agenda, but it is in the trend. 5-6 years ago, we worked a lot on data harmonization within the 3 EU agencies: European Chemical Agency, the European Food Safety Authority, and the European Medicine Agency, so that they would also be able to share all the knowledge, all the toxicological tests they were generating for their own dossier, and give them free also for the general, for the scientific community. So there was discussing with the agency, with also the stakeholder, the chemical industry, the pharma, and crop life industry, to see whether there would be something willing to work on. Now, a good concept that is something a bit to look at is the Science 2.0. If there was Science 2.0, this means there was a Science 1.0. And Science 1.0 was mostly about collision and a one-way interaction. So basically, the scientists would do is publication, peer review article, report, and hopes for the others to read it. Job done. We have a EFSA roadmap that's just been done. We have many things, documents of very high value that are being performed, but as soon as it's published, we move on. And that's the problem of Science 1.0. The Science 2.0 is to, after that, interact with the people, present the work you are doing, but also co-create the document you are doing with the related individuals. So you create a dialogue. It's more a discussion. There is also reason to have a Science 2.0. We have more people, so the more scientists that are involved in the field. So the argument of authority saying I have a PhD is not really making a difference anymore. We'll see this again. There is a lot more data and information. So there is the level of knowledge that is doubling every second. So you can imagine that you need also different tools to deal with this information. And we need more sharing and collaborating because we live more and more in the world of specialists. And this world of specialists forces you also to have more interaction and more dialogue if you want to do a proper Science 2.0 for policy. Very briefly, I walk you through a bit on the idealized roles of scientists when they interact with policymakers. So you have the pure scientist that in general does his own science. You can imagine the Nobel Prize guy, or men or women. At the end of the day, what they do is, it is not connected with the societal links. It's pure research. And it's only years afterwards that we understand that there is a benefit for the society. But the first impact at the beginning is just basic science. You have the science arbiter, and this is more the expert. The expert is being asked a specific question. The people that are sitting in scientific committees or consumer safety, for example, is typically these people where you give with a question that is framed, so you can answer by yes or no at the end. There they use their applied science to fit, they use the basic science to fit the societal needs. The third one is the issue advocates. These are the lobbyists. These are the people that are doing advocacy. They see science as a resources, and they cherry pick also the information to fit a specific policy option that mirrors their agenda. And the fourth one is the honest broker. And this is what, for example, the Joint Research Center is pushing towards, or at the European Commission level. They see science as a resource, but they don't cherry pick. They provide, based on the information, different policy options. In Europe, we call this a lot impact assessment. All right, so there are different models to look at. This is the linear models, and for the last two, it's more stakeholder models linked on discussion, co-creation. So that's to give you really very shortly, I will walk you through some of these principles that helps also to do the work at policymaking level. Now, us as scientists, we tend to stick to the facts, and facts are driving us. The problem is this: if we are only looking at facts, they work, but on specific files. For example, when you want to make earthquake detection, you need facts, you need how many earthquakes you are, what are the zones, and then you can develop a roadmap how to deal with earthquake. But if you deal only with facts, then it's a technocracy. So it's a place where only facts happen, although we know that people have values, okay? And this is something else to consider. But in a place where only values are taken in consideration, for example, we have abortion, people are pro-life, pro-choice. This is value-oriented, okay? If you only have value-driven, then it's populism. And it goes for different examples. But when you have facts and value that are taken together, then you might go towards democracy. And this is what we have, for example, with GMOs. We have some facts that say that it's useful, some values that tell us, okay, we don't want this model of industrial agriculture. So these are things to consider also when you discuss with policymakers, because your topic falls probably in one of those categories, or is a mix of the facts and mix of values. You should be aware of it and be able to frame it. All right, I'll walk you through three major challenges that I have encountered during these 10 years. There are much more. I could have made a top 10 list, but within the time I have, three are already good enough. Agendas dominate the policy process. That's the first time I heard these things when I was starting to work with EU institution. People have agendas. I had no clue what they meant. I thought notebook, okay, everyone has a notebook, but they mean they have a goal, but a hidden goal. And that's one of the ideas that we have. But at the end of the day, everyone has an agenda. And I give you a few examples, but what is important is that you understand what makes the people react to a specific file. So if you look at the European Consumer Organization, they want to defend the consumers. If you want to discuss with the animal welfare people, they want to defend animal use. They want to defend animals, and it's maybe more ethic-driven. If you look at the green NGO, they want to defend more the environment. And if you want to discuss with the industry, what they want is to put their product on the market. These are their agenda. They are very clear, they are obvious, but you need also to be in the mindset that the agenda exists for everyone. Policymakers do not understand science. That's the statement we regularly hear. First of all, I took this from VoteWatch Europe after the last term that shows a bit the background. You can see educational background in technology and hard science for all the political groups. So roughly, you have a quarter or a fifth of the members of the European Parliament that know something about science. They have studied the right things. They are not just lawyers or businessmen or etc. But you have a few that in general also sit in the specific committees that are more related to their studies or their values, so facts and values. So this is already to break one of these myths that we have around policy making, that MEPs are stupid, don't get anything, or that the policymakers don't understand the science. There is also a question of building trust, and scientists from the Joint Research Center are regularly going also at the European Parliament. For a while, they were doing shadowing for a week with an MEP's office so that they would follow also the political agenda of an MEP. And this helps also to understand what's going on the other side, because if you keep on living in islands, it's also not the right way to understand how the others are working. The third one, policymakers do not grasp uncertainties. This is a concept also that we live in. We live in a world of uncertainties. That's clear. And in general, scientists said, oh, since we don't know, we can't do anything. All right? And I always like to push the argument up to a maximum where if I have a scientist saying, okay, sorry, the level of knowledge is uncertain, so we cannot give recommendation. And said, okay, fine, I give you 20 years, 20 billion euros, come back to me. 20 years later, hoping that this person would reduce the uncertainties, and because this is what we want, scientists, always more science, more knowledge. We want to fill this gap. So I give you the money, I give you the time. Two things can happen. You find the evidence after 20 years, but most of you have done a PhD, guys. Huh? And you know what happened? At the end of the PhD, you have more questions. You end up in your discussion saying, okay, and we should do this and we should do that. So at the end of the day, it's a catch-22. More science for more evidence can be a vicious cycle. So at some point, you need also to stop. It will never end. And that's the difficulty for the policymakers, but also for us. For the policymakers, they spend their time dealing with incomplete set of knowledge. And they still have to take decisions based on facts and values. Whereas us, we consider that as long as we are not totally certain of something, we can't take a decision, but we have to learn how to do that. So sometimes also it's create more damage not to act. So we have also to be able to derive policy options and being flexible based on the political agenda and the timeline that is also imposed to us. These are ways to deal with uncertainties. The scientific advice mechanism from SAM, this group of chief scientific advisors, have provided different ways to do and deal with uncertainties. Here I'm listing how to do that. You can rank it: known, partially known, unknown, unknowable, and frame it. This helps also to drive the discussion, and we feel better, honestly, when we have something like this. We are not going also everywhere. I'm almost done. Here I wanted to tell you also, mapping the environment as a stakeholder is a very useful exercise. We don't do it enough. Who are the actors? These are in general the idea that's only one person who decides. That's absolutely wrong. Most of the time, it's a lot of people, not just one pen holder. It's a lot of people dealing with it. You need to understand what are the institutions, what is the institution leading the file, so you understand what will be the spin given to it. The policy network is particularly interesting. You can call it also the stakeholders or the pressure participants, because they want to influence also the discussion, and you would see maybe some of their wordings within the final text. Ideas that you want to share are at the beginning maybe a concept, but they can be at another level, values towards the person is more responsive. And the context is influencing everything. Over the last 10 years, triage was never a hot topic, never. When you look at political agenda, we had the Grexit, we had the Eurozone crisis, we had the bomb attack, we had the terrorist attack. Now we have the Ukraine war. I mean, this is 3 hours is not something that makes the people really driving the topic, but nevertheless, you could have some impacts towards your file. But understanding the context is really useful. Now what I'm showing you is, and I think it's my last slide, it's a legislative footprint. And I want to walk you through the mapping, just what I did before. And this is a file that was discussed 5-6 years ago during the glyphosate issue. So I give you the context. The glyphosate issue, the European Parliament had made a special committee just to deal with glyphosate. Context, issue. Now, who are the actors? It's 2 members of the European Parliament. So we are the European Parliament. It's MEP Norbert Linz, MEP Bart Staes, Green Belgium MEP, right-wing German Belgium, and right-wing German MEP. So they had agreed to list all the guys who went to visit them. And you will see also, the animation is not working. Wait a second. It is working. They are all doctors. They are all doctors. I told you, being a PhD, having a PhD is not making a difference. You are just one among the others. You see that you have the ministries coming, and the German ministries that are coming to see the German MEP. So you have a very local impact also with the policymakers. And here on the other side, it's the same for the Belgian MEPs. So you see the pressure participant, you see the institution is the European Parliament, you see also the ideas that probably based on the organization are being pushed forward. And I will stop here. These are further reading that I invite you also to have a look at if you want to pursue in the topic. I guess I was too long.
No, not at all, Francois. Thank you very much. It was a very nice, quick overview. I don't see yet any questions in Q&A, but I would like to ask you something. I know how important it is to express correctly what you are thinking. So do you think that we scientists are actually speaking a different language when we talk to the policymakers? Do we have to really learn their language to convince them about the need for the change, or how would you see it? What we need to do to be closer to them, to be able to convince them?
We need the way we are educated when we are doing PhD and science is to be rigorous, to be exhaustive, and to be precise. All these things are useful when you want to talk to the guys, okay? But if you start to talk to the way you are used to for publication, like you say in introduction, the material and method, the presentation of the results, they're gone. So you start with the conclusion. And this is where we are not used to, because in science, you first explain why people should listen to you, because your material and method and protocols are really robust and your statistical analysis is amazing, okay? But they don't care. So you need really to start by the end. And when we are doing all these science communication issue activities, I think it's a good way also to be ready to talk to MEPs, but still it's science communication, and in general, it's the target audience is still scientists. It's rarely the general public, it's rarely the policymakers. So we learn how to use tools, but still don't use them properly because of our education. So we need to work on ourselves. And me, I'm still need also, even after 10 years, especially when you are so much involved in a project and you know all the details, extracting from this very micro vision, going back to the micro, going back to the macro vision and the helicopter view and why we are doing that is always a challenge. And I always need a couple of hours or days before I can do this again. So, but it's feasible. It's just that I've been in so many meetings where the guy said, I have 5 minutes. And you had planned for 30 minutes. And you have to find your key message and you have to say, okay, I wanted to talk about this and this is the problem. Or this is something I want you to be aware of and something to monitor. Not easy.
So in fact, we scientists should be more trained in so-called elevator speeches, right?
In so-called
elevator speech.
Yes, it's a good exercise. But really, for me, the important is to start with the conclusion.
Okay. Good. I don't see any questions in the chat. So I would like to thank you, Francois. If anyone has any questions, please don't hesitate to write it and Francois will answer it personally. Now it's time to turn the floor to the next speaker, Dr. Kristie Sullivan. Kristie Sullivan is a Vice President of Research Policy within the Physicians Committee for Responsible Medicine, where she directs efforts to implement human-relevant alternatives to the use of animals in medical research, education, and the testing of drugs, chemicals, and other regulated products. Mrs. Sullivan received her Master of Public Health in Toxicology from the University of Michigan. Before joining the Physicians Committee, Mrs. Sullivan worked at the University of Michigan Occupational Safety and Environmental Laboratory, and the New York City Department of Health and Mental Hygiene. She's founding member and secretary of the American Society for Cellular and Computational Toxicology and has served on a number of governmental advisory committees. Kristie, thank you very much. The floor is yours.
Okay, well, thanks, Helena, and thanks to Francois for really setting this up, because I have to agree with most of what he says wholeheartedly. When you're talking with policymakers, you have to, what we often say is meet them where they are, which is what is going to speak to them, not what's important to you, what's important to them. But my presentation is going to be a little bit different today. It's about the reverse a little bit, how policies that are enacted affect the use of new approach methodologies in science. And it's also, a lot of this work is informed, like Francois, in the trenches, doing a lot of lobbying and policy activities over the last 15 years or so by my organization and my team and others. So most of you are aware with the motivations for moving away from in vivo toxicology. We need better tools to make more species relevant decisions about chemicals, and we need tools that give us better information so that regulators can make decisions and industry can make decisions about those chemicals. So there are public policy justifications for moving away from animal tests and implementing more advanced decision-making tools. And when I'm talking here, I'm talking about policies in very broad terms. So we can have legislative policies, laws, directions from legislators, or we can be thinking about corporate or institutional policies or practices, regulations, guidance, guidelines, or requirements that are implemented by regulatory agencies, and even policies and practices or statements that agencies make. Those are all sort of general policies that we can look to see how they affect the implementation of new science. They, in fact, may require some action, like a law or a legislative directive, or they may simply set the tone for actions that people take. And in our work, I have noticed that policies can inspire new science by setting a clear goal or providing funding to inspire innovative methods to be used. Policies can signal a direction, so that draws the field's attention and communicates priorities. And policies can stifle the implementation of new science. They can make it difficult for innovative methods to advance or be integrated into current practices. So maybe the most classical example is something that happened some time ago now, the EU Cosmetics Directive, which certainly led to a big increase in funding and motivation to develop and implement NAMs for safety assessment. And we also find that other sectors are benefiting from these investments, and we're seeing a lot of similar laws being advanced in other countries and states in the US. Another example would be the Lautenberg Chemical Safety Act. This was passed in the US in 2016. This law governs the US EPA's regulation of industrial chemicals. The law contains a number of requirements and provisions that facilitate the use of 21st century toxicology. Unlike the Cosmetics Directive, it doesn't set a ban, but it requires that the EPA do activities to reduce and replace animal-based tests, incentivize on EPA and industry to develop and implement alternative tests and approaches. And the law also required the EPA to publish a strategic plan and a list of NAMs. And so, of course, since 2016, there's been quite a lot of activity by EPA, not only in the relevant office, the Office of Pollution Prevention and Toxics, but also across the agency. We saw first the strategic plan mandated by TSCA published, and then that has now led to an agency-wide expanding beyond TSCA new approach methods work plan. And just this year, the EPA published a new chemicals collaborative research program talking about the work that they are undertaking, the science that they are undertaking in order to meet the requirements and the directives of the law. Oh, and just to mention, these links are hyperlinked, and we'll be sharing the slides so you can, my slides, and you can view them later. So as part of TSCA, EPA was also required to create and update a list of alternative test methods and strategies. They have done so twice already, and a couple of features I think that are really important about this list to emphasize is that it provides a starting point for potential NAMs to use for TSCA submissions. And it includes things like OECD test guidelines, EPA test guidelines, and other guidance documents. But it also suggests other tools and approaches like computational methods, integrated approaches that have been published by various entities like the OECD. Really important for me is that it clarifies that the EPA will accept other NAMs that are not on this list, and that the list will change to accommodate new science. And to help that new science along, it provides criteria for evaluating other NAMs that may not be on the list. So it gives some information for industry. So in fact, this document becomes a policy instrument to inspire the use of NAMs more frequently and the optimization of new approaches. So we also see that the European Union may start leading the way once again in inspiring new science to meet public policy needs, with its recent resolution calling for the phase out of the use of animals in biomedical research. I think this is going to be very interesting to watch how this plays out. I think what's very important is the details, right, of the roadmap that is put in place. Will it be strong enough to push the science forward? And how will scientists respond? A little bit of, I was thinking about this when Francoise was talking about decision making and sort of not being able to continue working further and further and further for the next 20 years, but say, okay, we have a policy goal, let's push the science in that direction in order to achieve it. And we can also see an example from the US Food and Drug Administration. They are making strides to advance implementation, spurred by stakeholders and by Congress, actually giving them advice and direction. So they have some reports and a work group about advancing new alternative methodologies at FDA, and part of that is also the ISTAN program, which is meant to approve or qualify human biology-based methods for use in drug development. So this has become a priority of the agency because of policy priorities and stakeholder priorities. So signals. How do policies send signals? One really good example is found in the US EPA Office of Pesticides. So they have been publishing some policy documents. You can see on the bottom of your screen, get this going here, over the years to provide for specific opportunities for non-animal approaches for specific endpoints. But in 2016, we also saw EPA publish a letter to stakeholders on EPA's goal to reduce animal testing, specifically for the acute six-pack of endpoints. And I also wanted to mention, actually, since 2020, there has been additional progress on some endpoints, but official policies haven't been published. So we can see the results. EPA provides metrics on its website. And for example, we saw the potential to waive the acute dermal toxicity test for formulations and single chemicals. And we can see the industry responding and submitting more and more requests for waivers to avoid that test. And of course, they published a policy accepting in vitro skin sensitization approaches, and you can see year by year a big jump in submission of those approaches. And while the publication of explicit policies outlining the acceptance of NAMs is best, even in cases where explicit policies have not been published, for example, in skin and eye irritation, they have not yet put out policies specifically related, except for antimicrobial cleaning products for eye irritation. But you can still see a big increase in vitro eye and skin irritation assays. And I should say this could also be a result of EU or UK policies to request in vitro data from industry where those methods exist, and then the industry sort of conducting both in vivo and in vitro and submitting the in vitro to EPA. But I think also we can conclude that the letter to stakeholders has communicated to the community EPA's direction, and stakeholders are taking that direction. Another example from the Food and Drug Administration. A few years ago, we analyzed new drug applications submitted to the FDA. And one of the things that we found is that for irritation, there are a fair number of in vitro or ex vivo eye irritation studies submitted. And that's not the case for skin. So I wondered why that might be. And it turns out that there is a recent FDA guidance in 2015, relatively recent, that recommends NAM use for a certain subset of products. And the guidance actually states that in vivo rabbit ocular irritation is no longer recommended for topical drug products. So we have here, in cases where agencies put out policies, the signals are taken up and industry uses them. So the bad news is that policies can also stifle science. FDA is not required by law to require animal tests. However, they do have some regulations that specifically state, the effect in animals. So that's an explicit requirement for animal tests. It would be fairly straightforward to neutralize this language, to go through all the past guidance and change it to non-clinical rather than animal. And this would allow for animal data to still be accepted when that's when the FDA feels that's needed, but also opens the door to using NAMs where appropriate. It there has been some activity in Congress on this. Congress has put forward funding bill language in recent years directing FDA to update its regulations, but that language is only a preference of Congress. It's not binding legislatively, so we will continue, I think, to see activity here. There is also some guidance that FDA has released, in particular this non-clinical guidance. This is an ICH and FDA guidance that says methods may be used to replace current standard methods. But unfortunately, that then comes back and says you can use an alternative approach if it satisfies the requirements of the applicable statutes and regulations. So then we go back to the regulations and see that those are still pretty specific related to animal tests. So it's good that it provides flexibility, but there's some more clarification that's needed. So I also wanted to just very briefly address the European Union REACH and CLP legislation. I think that we've seen over the years that the intent to reduce the use of animals in chemical testing in the legislation has, and ECHA and OECD have done quite a lot of work to push forward some alternatives, such as the QSAR toolbox, a lot of activity on guidelines and refining activity in read across and refining the process of read across to make it work better. However, I think that we have seen that very strict data requirements within the tonnage bands erect a high barrier for using new methods or flexibility to accept alternative approaches. We have heard directly from regulators in the EU that unless a method is fully validated, OECD test guideline within the regulations, they can't accept it. And that often is sometimes difficult to do or takes a long time. So I will close now because I'm really looking forward to the next presenter as well. But basically what I've tried to tell you is that I think that policy should reflect our societal values, and that updating and strengthening policies can go a very long way towards inspiring and facilitating scientific progress, but that it is required to ensure application of new science. And I've learned really statements are not enough. It needs to be also supported by funding, actions, and changes. With modern policies, we can get modern science. And I will close and thank you for your attention.
Thank you very much, Kristie. There is one question in the Q&A, actually for both of you, Francois and Kristie, from Sue Leary. What is the role of internal advocates, e.g. in these very large agencies or legislative committees? There can be people who are receptive to your message, but not the top policymaker themselves.
Yeah, this is a really great point, Sue. I think when we think about change at the agency level or at the policy level, these things take many years. And so we do see over time that advocates within these large organizations really help to push activities forward and help advocates and help policymakers continue to try to make progress throughout this long timeline, because of course we've seen many years of activities, for example, at the EPA level, build upon each other to lead to the progress that's being made right now. Great question, Sue.
So would you like to add something?
Yeah, two things. Thomas Hartung used to say, if fish stink, it usually starts from the head. So that gives you the image. Having internal advocate is always good. It's good that you feel that you are listened, considered, but it's not, depending on the food chain, how, because we are talking about very hierarchical entities. And even though if you have people that are at desk officer level, that doesn't always make a difference. So you really need to have also leaderships toward those things. We see it with the Food Safety Authority, and they don't have a proper mandate to develop NAMs, although they are putting 20 million euros for only this year's on the development of NAMs. I'm blasted when I see this. And they go beyond what is written in the regulation about EFSA. Now you can guess what I think about what the effort that ECHA is doing on these activities.
Well, thank you very much for answering this question. I don't see any other questions, and for the sake of the time, we will continue to the third presentation that will be delivered by Dr. Nataliia Bubalo. Nataliia worked at the Medved's Research Institute or Research Center of Preventive Toxicology, Food and Chemical Safety in the field of general toxicology from 2008 to 2018. Nataliia established GLP laboratories of alternative research methods, implemented in vitro methods in the center, and created an Ukrainian 3R center. Nowadays, she's working at the National University of Food Technologies as head of the course Basic of Toxicology, and in Yacht Life Science Consulting as toxicological expert. She works with the Ukrainian government to implement new cosmetic legislation which bans animal tests. Nataliia, I'm very much pleased to have you here today, and the floor is yours.
Well, thank you very much for your kind introduction and that you invite me. It's a pleasure to talk today, and last but not least speaker. It will be interesting what will be the next my slide, and I think we will have next interesting discussion. So let's start. So my first phrase which I want to say is that we are scientists. Our responsibility to make sure that policymakers understand the value of our science. Maybe we will have then some what to add. And here it's like science generate knowledge, scientists prove knowledge, spread it, and make knowledge understandable through education. Then society accumulate knowledge, and we have effects in a policy. On the slides, process looks very simple, clear, but in the real life, it takes a lot of force and time. And as my previous speakers explained it, a lot of many really force we need to put to have so clear and step-by-step process. We saw that one of the important things in this diagram or in this process, as from my side, it's making knowledge understandable through the education. And I think it's the most effective tool. Today I will be more precise, and I will talk about alternative methods and how it works in Ukraine, and just share how it exists and what we are doing in our country with this scientific knowledge, how we try to implement it in our legislation, in our testing, and so on. So today I would say that for scientific side, the process of implementation was consistent and successful. In a policy field and like in a legislation level, it is continued, and I would say it's not the first step. But we have some positive moments, and one of the positive example I want to share with you this Ukrainian law. And Ukrainian has a huge pesticide market, and it is very important that policy, legislation, and last scientific knowledge works together. The policy reflects science, and I saw that it's not only my opinion and my previous speakers too. And it is an example of Ukrainian law which implement EU legislation. And here you see like Ukrainian law and how it's implement and work. So when we implement this EU regulation 1107, it gives us the possibility to apply principles which are mentioned and in the Commission Regulation 200 or 284, 2013, and we are applied the following principles, like a triad approach in testing, priority is given to in vitro methods, and reduce animals in the studies. And it is implied, and this has already, these principles already applied in Ukraine. First, how it's work. In Ukraine, we have specific scientific commission on integrated hygienic standardization and regulation of pesticides and agrochemicals. And this commission have a discussion how we can apply this legislation in Ukraine and work with it. After it, we go further and go to discussion with the government, and in this case, the government body represented will be a Ministry of Health. Then Ministry of Health goes to the Parliament with this idea to provide this legislation into the law, in Ukrainian law. Again, discussion committees, and at the end, the Parliament vote. We have legislation appears and the law come in force. Of course, it's like easy in this hand, and we try to be as much as effective as we can, but at the end we receive it and can work, because for our side, like people who respond to make at the end all investigations, we now can use this approach, this in vitro methods to reduce using animals and to have a priority to in vitro. Another one positive fact of implementation in the in vitro test made it possible to adopt a new regulation of cosmetics products in Ukraine. According to this legislation, testing cosmetics on animals is forbidden. This law should or will become in force in August 2002, but unfortunately, this deadline will postpone it. And you know what is the reason. We know this in EU, this legislation became in force in 2009. It has 5 years to implement it and to achieve the main goal to ban the test cosmetics product on animals. The changes were possible in Ukraine because Ukrainian toxicologists have already introduced alternative methods. In fact, with the adoption of the regulation, European regulation is being implemented in the Ukrainian legal field. But one important thing which we need to improve, and it is a scientist's duty, is a scientific committee for cosmetic regulation. We definitely need this tool to it's not exist right now, but it's essential for a better interaction between last scientific knowledge and understanding in the policy market and regulators and subsequent implementation into the law. Otherwise, all of the steps cannot be done because the most important are not exist. And we are actively working on it just to be clear and sure that we can implement this law in a correct way and use it appropriate. But all changes and all goals which we achieve can appear in Ukraine because of existing this center, the State Enterprise Medved Research Center of Preventive Toxicology, Food and Chemical Safety, Ministry of Health. It's a scientific and research center with more than 50 years experience in the toxicology. And this is a place where all toxicological field apply. It's only one in Ukraine, but it's enough and enough huge to cover all challenges which appear in the toxicological field in a so big country like Ukraine. A wide range of toxicological study for risk and safety assessment of chemicals is conducted in this center, and you can see it on the slide. 300 people work there, and specialists from the Vet Research Center were involved in the regulation process and as a consultants. Also, scientists, they are in the scientific committees, and I think most of or some of our staff or colleagues will be invited and organize this scientific committee for cosmetics. So we have GLP certification and ISO certification. Our GLP lab have this certificate more than 10 years, and we apply all of existing in vitro, which not all, but most of all, we try to apply a special OECD test, which is in vitro in our lab. And here was born a Ukrainian Research Center, and scientists from different fields, not only from not only in vitro specialists were involved, and it was not. We have some reason why to do it in this way, because we are working to apply all three pillars, not only replace, but also a reduction and refinement, because we're strongly involved in the regulation process in our country, and from regulatory purpose, not always we can use only in vitro methods to completely fulfill all questions which appear. And here it's very nice to use reduction and refinement to improve these principles and to work better, to definitely use the latest scientific knowledge and to be on a safety level. We are collecting knowledge and share, spread, and educate among the target and responsible person and bodies. This principle should be understandable and actively promote among involved person and the society. At that moment, it's a main task for the Ukrainian Research Center, and we are thinking that the best way to achieve this goal is it through education. And here we use another quite good tool. It's an important cooperation between research center and the university. To spread a knowledge, and here became to the first my slide where I said that it's our responsibility to make for us like a science to make knowledge understandable. We share it through university, but also we have both benefit. Sharing information with the university and connecting between two government branches like Ministry of Health and Ministry of Education. And when we are going to the parliament and when we need to implement something, we have additional supportive tools which can work together. Of course, it's not like today we make a decision, tomorrow we have results. No, it took more discussion, lots of letters, lots of force. But we try to have the most effective model, how to use it and to implement our scientific knowledge. And in this university, we have toxicological course. And in this toxicological course, we have a model which is talking about the alternative methods and 3R principles. And why we use especially this university like one of the two? Because the participants of this university will be the future technologists of cosmetics products and food products. So it will be people who will work on the manufacturers, who will work in governments, who will work in the regulation field, and they have already got all necessary knowledge, and we can speak on the same level to understand the reason why to do it. Also, we have like toxicological course for specialists, and here our participants, it's people from the government, industry, regulators, and scientists. And here again, we can spread our knowledge to the targets bodies, to the targets people who at the next will go and work on it. And we don't have this barrier. Yes, it's not always works perfect, but according to our experience, I think it's one of the best way how to do it and to improve it and reduce time, because always anyway we will achieve it anyway. Now we saw how it's changed in United States, how it changed in Europe, how many in vitro methods, alternative methods has this OECD guidelines, can be used in the regulatory field, and regulators, authorities can read and this report and can said yes, we can use it. But anyway, if we can make it quicker, why not? Just to use appropriate way and to use all tools which we can use. And my end slide will be unusual, and I will thank not only for your attention. I want to say thank you for all support, and for each person. I want to thank a big thank you for organizers, and also for, and I write toxicologist for toxicologists, and my personal thank you to two toxicologists. We support Ukrainian toxicologists, and not only me, also a toxicologist from Ukraine, and to Helena Kandarova, because she did it and she support two Ukrainian families, toxicologists, and share with them their apartments. And I'm very proud that I met this person in my life, and it's a big pleasure to know you and to have the same goals. We will achieve you. And also I want to say thank you to Arneiaks, which support me and my family, and I can stay here and to continue to work, to continue to improve my knowledge in toxicology and all my force, which I've done before, and to continue it. So thank you. Be brave, stay with Ukraine.
Thank you very much, Natalie, also for the personal words. I mean, I was in your institute in 2019 and was very impressed by the level of the organization and as I wrote also equipment and overall policies that were implemented there. And I know how difficult it is to implement some new policies, especially in the situation that we are, for instance, not yet in the European Union. We went through this also as Slovakia when joining European Union. I wanted to ask you what are the complications or difficulties in implementation of these European laws? Is it easy, or is there some, hesitation whether these methods, new alternative methods, are good enough to replace what has been established in the past?
So, as I said, it's in the scientific field, everything is clear, understandable, and no questions. But again, here we need to be on the same level with the regulators, and especially with persons who really apply this law. And in one case, especially with cosmetic regulation, in my opinion, like a scientist which use this law every day, it's quite clear and understandable. But here I saw that we need to educate people to understand how to use it law appropriate. Especially in this case, we need a risk assessor, safety assessor, who can create a scientific safety report in cosmetic things. And it's not a problem. It just to take a time. We know how to spread. We have this knowledge, and we are lucky in it, because otherwise, if Ukraine will not didn't exist this institution, that's why I mentioned about it, because it's the core of toxicology, and it's very important in nowadays because it's a barrier. It's a shelter which can protect people and be in a safety. Otherwise, it will be a big problem. And the difficulty, maybe it's explain that toxicologists, it's very important because here we have some gap in steps, because in Ukraine, toxicology not exist like general toxicologists, only clinical toxicologists exist. And we don't have this degree like in EU or United States. And now we actively work on it. That's not only medical doctors can be a toxicologist, also biologists and chemists, and they apply this course, and then a master program, a Master of Science program, just to appropriate educate and have this more people, because we need more stuff. And in an appropriate level, and it takes at least 3 years to be like understand something. I think in this case it can be a problem, but we are lucky that we have only one institute which is working this field, and like if we agree with it, we definitely agree everywhere, and it's like no discussion, but we believe that we are on the right level of scientific level, I mean, to prove it.
Well, thank you so much again, Nataliia. I don't see any other questions in the chat, but I would like to conclude maybe with what you said. Education is really the key to the success. So we need to educate each other and train each other in proper speech and soft skills to be able to express what we mean and what we think, and to be able to collaborate. And as Francois said, the science to zero, and this is something that runs the both ways. So I hope that if we are educating each other and improve the communications, we will also see accelerated progress in the implementation and use of the 3 Rs. I thank very much to all of the speakers. It was a brilliant session. Thank you also. Big thanks also to everyone who joined this session today. And we wish you a nice evening. And as announced, this session will be available in the archive for all those that didn't manage to see it. So thank you very much and have a nice day. Bye-bye.
Bye-bye.
Thank you. Bye-bye.